Hello students, now we will start learning about Metaphase 1 onwards till completion of Meiosis II and Cytokinesis 2. So first one, Metaphase 1. Let's draw the diagram. This is a cell and here will be centrioles because we are discussing about animal cell. So I am giving out centrioles, asteroids and chromosomes aligned on equatorial plate. So this green color here and this red color here, small little parts of the chromosomes are nothing but the exchanged genetic material during crossing over of packaging. So now centrioles have asteroids. On the kinetochores, the spindle fibers will be attached to either poles by attaching spindle fibers to the kinetochores of chromosome. So this line where chromosomes are aligned is called equatorial plate. Equatorial plate. So this will be the chromosomes aligned. Now we'll write some points regarding this. Both chromosomes are aligned on equatorial plate. Now, in Metaphase 1, chromosomes are said to be attaining prominent structure and morphology. So chromosomes become more prominent. Means they are clearly visible, so that at this stage we can study morphology or structure or shapes of chromosomes. So this Metaphase 1 is said to be the best stage to study chromosomal structures. Best stage to study chromosomal structure. So we will give some of the labelings. Here the spindle fibers that are attached at one end to the centriole, other end to the kinetochore. These we can also give a name like discontinuous spindle fibers or chromosomal spindle fibers. And the type of spindle fibers that are connecting two opposite pole containing centrioles are continuous spindle fibers and also said as interpolar spindle fibers. So these are important points of Metaphase 1. In Anaphase of mitotic division, we have seen there is a splitting of centromere of the chromosome, and so that chromatids are moving to opposite poles. But here there is no splitting of centromere, but chromosomes completely they are moved to opposite poles. We will draw the diagram. So here, during Anaphase, the chromosome completely itself has moved to opposite end. No splitting of centromeres. Chromosomes are dragged towards opposite poles. This dragging of chromosomes towards opposite poles is given a term like disjunction of chromosomes. So, existed two chromosomes of the homologous pair will be moving to either side of the poles. Now, here in complete cell, there are two chromosomes side by side as homologous pair. But now, now the condition we can say it is 2n condition. But now they started getting into opposite poles so that later they will be converted into half of the chromosome in this part and half of the chromosome in other part. So that is said as conversion into haploid form after cytokinesis. So chromosomal number starts to get into half the number. Now, in metaphase, in mitotic division in anaphase, we have seen formation of interzonal spindle fibers. But in case of anaphase 1, there is no existence of or formation of interzonal spindle fibers. So interzonal spindle fibers are between those chromosomes which are moving to opposite poles. Here we don't see formation of interzonal spindle fibers. Next one, telophase 1. Telophase 1. In case of telophase 1, if you see the diagrammatic representation, we have seen before in the differences between mitosis in animal cell and mitosis in plant cell. As if the formation, the cytokinesis that happens in case of animal cell is after anaphase, but in case of plant cell, cytokinesis happens after telophase or at the end of telophase. Now, so if I write here about invasination, it means that we are talking about animal cell. But here, constriction at the peripheral regions or at the borders, that we call as invasination. So what are other changes happening? The chromosomes that have been moved to opposite poles, now they will be decondensed to chromatin. There will be formation of nuclear membranes. So that there will be formation of other cell organelles also. We'll write this point. Cell organelles, nuclear membrane, nucleolus reappear. But here we are seeing spindle fibers, but here I did not mention any spindle fiber. Spindle fibers disappear and chromosomes will be decondensed, or they will be recoiled back to chromatin material. So these are the changes that are happening up to telophase 1. Then what about cytokinesis? So we'll see cytokinesis 1. During cytokinesis, if it is an animal cell, cytokinesis will be by the process of invasination. So here this invagination will be protruded towards the central region. So the type of cytokinesis is centripetal type. Centripetal type of constriction, which is moving from the peripheral towards the central region by invagination process. So this part will be separated. In the next one, so the constriction has been means increased much more. Now two baby cells will get separated. They give labelings for this. These are two daughter cells. This one and this one are cells. This is chromatin material. This is chromatin material. Here the condition is 2n. They got separated. Means they started moving to n condition, haploid condition. So here also it will be haploid condition. So here also it will be haploid condition. Here also it will be haploid condition of each daughter cells. These are daughter cells are produced. We'll write one point. So after meiosis 1, chromosomes, after meiosis 1, the daughter cells will get. Two daughter cells receives half the number of chromosomes. From parent cell. In parent cell, there will be diploid number of chromosomes, but after formation of after cytokinesis 1, there will be haploid number of chromosomes. So this is up to cytokinesis 1. Later, the cell has to enter into meiosis 2, but between the time period of cytokinesis 1 and meiosis 2, we also can use a term like after cytokinesis 1 to karyokinesis 2, because again in meiosis 2 there will be division inside the nuclear material and in the nucleus. So we also can use a term like after cytokinesis 1 to karyokinesis 2, there will be some resting time period or resting phase that is very much short, called as interkinesis. Now we will see about those steps. After cytokinesis 1, before entering meiosis 2 or karyokinesis 2, cell has short resting phase called interkinesis. Now starts meiosis 2. Meiosis 2 includes prophase 2, metaphase 2, anaphase 2, telophase 2, and the last cytokinesis 2. So first one, prophase 2. We have got two baby cells in meiosis 1. The condition of chromosomal number is haploid in condition after meiosis 1. Two daughter cells with haploid nuclei now starts meiosis II. Meiosis II is equal to mitosis or mitotic division. We said here two daughter cells. We'll write first daughter cell that received female chromosome from homologous pair. So we'll write prophase 1, prophase 2, not 1. So prophase 2. About the nucleus cell. Here we said that it has received the female the chromosome from the female gamete, which has underwent crossing over during packaging. So crossing over starts in packaging, ends in diploting. Here are central. In prophase 2, what happens? All the nuclear membrane cell organelles, they start to get disappeared. So this is first daughter cell. We'll write here about second daughter cell also. Second daughter cell. Here we should write like so here also we'll write received chromosome from male gamete from homologous. Here we will draw prophase 2. This is one daughter cell, so other daughter cell. From the beginning we are mentioning the chromosome from the male gamete with red color, so now we will write that only. This is the part or the genetic material that was exchanged during packaging. This is disappearing nuclear membrane. So cell organelles, nuclear membrane, nucleolus disappear and spindle fibers, aster rays reappears. Here also the same thing. Cell organelles, nuclear membrane, nucleolus. So this is cell. Disappear and spindle fibers, aster rays reappear. So next stage, metaphase. So during metaphase, this chromosome will be aligned on equatorial plate. So here also we will write about metaphase 2. This chromosome, now it will be aligned on equatorial plate. At this stage, we can easily read the morphology and shapes of chromosomes. Kinetochore, spindle fibers attaching to kinetochore, continuous spindle fibers, continuous spindle fibers. So in metaphase, we see only arrangement of the chromosomes on equatorial plate. Now, Anaphase 2. In Anaphase 2, we see splitting of centromere, appearance of interzonal spindle fibers, like mitosis, same. Same in Anaphase 2 here also. Here I don't have place, but all these points are applicable here also. Anaphase 2. Right, same. When you go for diagrammatic representation, here centrioles, aster arrays, there is splitting of chromosomes, centromere at a chromosomes at a centromere. Okay. So when it was cut in half, this crossover genetic material exchanged chromatid part will come to one side, other which did not exchange genetic material will move to other side. So we'll see, as usual, continuous spindle fibers, discontinuous spindle fibers, and also interzonal spindle fibers. This same one I will write here somewhat clearly. Centrioles, aster arrays, continuous spindle fibers. This one also will get broke up at centromere area. So the one which did not exchange genetic material chromatid, that part will be moving to one end. So the chromatid part which has exchanged genetic material will be moving to opposite end. So these are discontinuous spindle fibers. This is interzonal spindle fiber. Interzonal spindle fibers. So again I am repeating, splitting of centromere will be seen in anaphase of mitosis, anaphase 2 of meiosis II part. Next will be telophase 2. So at the end of anaphase 2, the invagination means the cytokinesis starts. By the time of telophase, the chromatid part which has been moved to opposite poles during anaphase 2 Now it will get decondensed into chromatin, which is surrounded by nuclear membranes. Now the cell organelles also starts forming. Here we should mention some of the exchanged genetic material part. Like this. So in vagination it will be moving. Centripetal type of cytokinesis will later during cytokinesis it will happen here. So in this also Telophase 2. Now what happens if this is the cell? Now the exchanged genetic material during puberty. It will be moving to opposite course. Now we will write the ploidy condition. Now when it is entering into meiosis II, the ploidy condition of the chromosome is N. Here also N. Here also N. 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 It is N. Now it is giving out 2 daughter cells. Two daughter cells. Now it is N. N. This is N condition. It also gives rise to 2 daughter cells. Inside the nucleus. So we will write how many haploid number of daughter cells have been obtained. N and N. If we come to count 1, 2, 3 and 4. So we have seen formation of 4 number of haploid daughter cells by the end of meiosis II even after cytokinesis. Since this is animal cell, here I have written by invagination, and if you want to write cytokinesis 2 is centripetal in animal cell. In case of plant cell, it will be by formation of cell plate, and that is also called as centrifugal type of cytokinesis after meiosis II in plant cell. So if you have place here, I will mention somewhere. Cytokinesis 2 is centrifugal in plant cell. In plant cell. So cytokinesis 2 is centripetal in animal cell, centrifugal in plant cell. So this is about meiosis I and meiosis II, in which we have obtained 1, 2, 3, 4 number of haploid daughter cells.